Welcome back. We are on our last packet for Module 4, Infectious Diseases, which I love. There was one point where I wanted to be an epidemiologist, someone that studies diseases, but now I'm a teacher, which I couldn't be more happy about, honestly. So today we are going to talk about bacteria. We're going to talk about gram stains and why they are important. We'll talk about classifications of infectious diseases and also viruses and HIV. And of course, we're going to throw a little bit of COVID in there. So, our question of the day is, how are viruses used to cure disease? For the pre-lecture, the main thing you need to know is this table down here. So we're going to talk about bacterial structures and their functions. So the first one is the cell wall. All bacteria have cell walls, and this protects it and usually contains a molecule called peptidoglycan. And this allows for classification between gram-positive and gram-negative, which we'll get to in a minute. The capsule is the gelatinous layer outside of the cell wall of some bacteria. And so this makes it lack surface proteins, so the immune cells do not recognize the bacteria as foreign, which is good for the bacteria, not so good for you. Next is flagella. So on our picture here, what have we talked about so far? The cell wall, and then the capsule would be outside the cell wall. And you have the flagella. This is for motility, so it can move. And it spins in 360 degrees. How do I show this? Like this. Whips in circles to help it move along. The fimbriae are stiff fibers that allow bacteria to stick to surfaces like host cells. So to attach to your cells and also to stick to other things like dirty doorknobs. So in our picture here, these little hair-like structures are the fimbriae. A pilus is an elongated hollow appendage used to transfer DNA from one cell to another. So in this picture, it's this right here. And bacterial cells don't really do sexual reproduction. It's asexual for the most part. But with this pilus here, they can transfer parts of their DNA to another bacterial cell. And last of all, we have a plasmid. Plasmids are really cool. In a bacterial cell, it's prokaryotic, so it doesn't have a nucleus, so the DNA is just kind of strewn about inside. But they can have these little circular DNA, portions of DNA, and a lot of times these genes allow for antibiotic resistance, and they're found in the plasmids, and that's one thing that can be transferred from one bacterial cell to another. So for the exam, just know if you're given the function that you can name the structure, or vice versa. If you're given the structure, you can name the function. So this might be a good flashcard way to study on that one. And next we'll get into gram-positive and gram-negative. This is bacterial classification. And what a gram stain does is it detects the thickness of a cell wall. And if you ever take microbiology or have taken microbiology, this is a very simple explanation. But gram-positive bacteria have a thick cell wall. And gram-negative bacteria have a thin cell wall. So again, I apologize, this is not in color, but gram-positive bacteria will stain a really dark purple color. And think of it, because it has that thick cell wall, it's able to kind of hold on to more of the dye, and it'll turn out being a dark purple color. Whereas the gram-negative bacteria stains light pink. Because it has that thin cell wall, it can't quite grab onto as much of the dye as the gram-positive can. And the reason we even have this type of stain on bacteria is gram-positive and negative bacteria have different treatments, and so you want to know which type you're dealing with, because you're going to treat the infection differently depending on that. So that brings us to antibiotics, and what they do is they inhibit bacterial growth. So it's not going to kill the bacteria that's already there, it's just going to inhibit the growth and reproduction of it. So there are a lot of antibiotics that end in "-cillin", like penicillin, ampicillin, I think there's even methicillin. And what they do, the mechanism of action, is they interfere with the cell wall growth, so more new bacteria can't be produced. So the amount of bacteria doesn't increase. Instead, those that are there will slowly die off, and then the infection can go away. So kind of random here, why do we refrigerate things? We know we can get food poisoning from bacteria on our food, and so we store it in the fridge. And what refrigeration does is it only slows the growth of bacteria. It doesn't stop it. So when you make something cold, it kind of slows everything down. So it's going to slow down the growth, not stop it. And then if you were to take that same food that has bacteria on it and take it out of the fridge and let it sit out at room temperature, that bacteria is going to start to reproduce, and that's why it's reproduce faster. And that's why it's important to always keep your food in the fridge and not leave it out too long. Okay, on to viruses. Viruses are very interesting, and in seventh grade, we go over the characteristics of life. There are 7 that we go over, and that's not a solid number. And it's even debatable among scientists, but I like to give them the classifications of life and then show them viruses and have them debate on whether they're living or non-living. And different sources will say different things. But for this class, just know that viruses are generally considered to be non-living because they're not composed of cells. And that's like the number one thing we teach with living versus non-living is all living things are made of at least 1 or more cells. And viruses are not composed of cells. Another one of the characteristics of life is metabolism, the ability to acquire nutrients and get those to make energy. And viruses cannot do that on their own. However, they can do that with the help of a host. So cannot acquire nutrients because they need a host to do that. So for the structure of viruses, they have a capsid, which is kind of like the outer container of the virus. And it's made out of protein. So protein container for genetic material. And you also need to know spike proteins. And this helps them bind to the host cell. So this is the influenza virus here, and you can see its spike proteins on the outside to help it attach to the host cell. Something else cool about viruses is their genetic material. It can be DNA, it can be RNA, it can be single-stranded DNA, it can be single-stranded RNA, it can be double-stranded RNA like ours, or it can be double-stranded, wait, did I say that right? Double-stranded DNA like ours, or double-stranded RNA unlike ours. So has some interesting genetics going on there. And are antibiotics effective against viruses? So a lot of times you'll be sick and go to the doctor and think you're going to come home with medicine that's going to make you feel better. And they'll say, sorry, you have a virus, go home, rest, drink plenty of fluids. And the reason for that is the antibiotics work on the cell wall of bacteria. And do we see any cell wall? No, because they're not even cells. So antibiotics target bacterial machinery like the cell wall. Whoops, I'm just saying antibacterial, same thing as antibiotics. Target bacterial machinery like the cell wall. Can also attack ribosomes in the bacteria. And so even if you're taking antibiotics for a bacterial infection, you can still get sick with a random virus. Because it has no effect on them. Stay. All right, on to, oh, just a quick thing for the exam. No, we'll do check mark. Gram positive and gram negative, and what color they stain, and whether or not they have a thick or thin cell wall. And for viruses, these are the main two things you should know. So classifications of disease. You've probably heard these words: epidemic, outbreak, and pandemic, and they all mean different things. So an epidemic is when a disease has more cases than expected for a period of time. So, for example, if a disease normally has 10,000 cases per year, but on a particular year it has 20,000 cases, that's twice as much, we would consider that an epidemic. An outbreak is an epidemic confined to a local region. So, like to one particular area. So an example of this would be like Ebola outbreaks that you hear about. In West Africa in 2013, there was an Ebola outbreak, and more recently there have been measles outbreaks that are just confined to certain areas. Like I don't know if you've heard about the measles outbreak in Disneyland, but yeah, confined to a certain area. And then a pandemic, which is the word you've heard a lot over the past couple of years, is global, a global epidemic all over the world. So, which one of these describes COVID-19? That's a pandemic. So, current pandemics that we have as of when this was made is COVID-19, HIV and AIDS, tuberculosis, which is a lung disease, and malaria, which is spread by mosquitoes. So the life cycle of a virus down here, and we've got a picture of it above. Step 1, 2, 3, and 4. Step 1 is spike proteins attached to surface proteins on host cells. So this big guy here is the host cell, and these are the viral cells. So step 1, they attach to receptor proteins that match up on the host cell, and they fit like a lock and key. So spikes fit on cell receptors like lock and key. Step 2 is membrane fusion, which you're seeing here. So the membranes of the viral cell and the host cell kind of fuse together, and that now allows the contents of the virus to enter the host cell. And step 3, viral DNA is made into, or viral RNA is made into viral DNA. And we're used to seeing DNA go to RNA, so we call this reverse transcription, and it combines with the host DNA. So we've got the reverse transcription here, RNA turning into DNA, and then it goes in and combines with the host DNA inside the nucleus. And then step 4, viral proteins and genetic material are made by the host's machinery. So new viruses assemble. They bud off and infect other cells. So here it is. It now has the genetic instructions to make the parts of the virus. And so it will start to build those. And once it's ready, it will bud off and run off to hijack another cell. So basically what's going on here is the virus is hijacking the machinery of the host cell to make more copies of itself so it can spread to other cells. The picture that you see here, some viruses have like different genetic material and different life cycles. This is just a general one, but this is what applies to HIV, COVID, and influenza viruses. Okay, so if we are designing an antiviral drug, what part of the viral life cycle would you want your drug to inhibit? So you can pause this for a minute and just come up with some ideas. How could you stop this virus from reproducing? I'm going to pretend that you paused it. We'll come back. So many antiviral medications target the virus's ability to attach to a host cell. So if it's not able to do that, it can't fuse with its membrane and get its contents inside to hijack the cell. It can interfere with reverse transcription. Turning that RNA into DNA. If it can't do that, your cells can't use the viral instructions to make the parts to make more viruses. Or you can prevent it from integrating with the host cell's DNA. So maybe it gets in, it does this reverse transcription, but it doesn't allow it to integrate with the host cell's DNA, so it's not going to make more viral copies or parts. And maybe as you pause this, you came up with some other innovative ideas that may work someday. So maybe look into that and roll with it and become, what would you even call it? An epidemiologist? I don't know. But these are the three you need to know for the exam. So, question of the day, how are viruses used to cure disease? So there are two examples you need to know. And this picture right here is just a general idea of how gene therapy works via viral vectors is what they're called. So if you can insert a therapeutic gene into a viral vector, so you've got a virus and you put the gene that you want inside there, and then it attaches to the cell surface, enters in, um, and then that DNA will incorporate itself into the patient's genome and will make the proteins that that patient is lacking or whatever it is that they need. So that's just a general idea. But the two that you need to know for the test are this first one, epidermolysis bullosa, and I think this is the third time we've brought this up. We talked about it in the whatever chapter we were talking about the skin in. And we talked about it when we talked about epithelia. So sometimes called EB. So remember, this is when the patient can't form their own basement membrane, so their skin can't ever really attach to their body, and it kind of just peels off. So with this, let me add, this is a basement membrane gene defect. So this is a patient, most of their skin is damaged. They take skin cells from the patient and they put a working gene that is the correct version of that gene to form the basement membrane into the cells. It will incorporate it into its DNA, and then they can actually grow these corrected cells into sheets of skin and then graft them onto the patient, which is really cool. If we could figure this out for a lot of diseases, and I think they have, that would be amazing. This is really cool stuff. This next one is sickle cell anemia, which I believed we've already talked about. When the person has a mutation where they don't form the normal round bowl-shaped red blood cells, and instead they're shaped like a sickle, like you see down here. So how this works is when the blood-forming cells, blood-forming stem cells have mutations in the gene for something called beta globin, they produce red blood cells that can become hardened and sickle-shaped. And you can imagine this can cause a lot of problems. They're not flexible, they can't fit as well through capillaries. So gene therapies for sickle cell disease aim to remove these stem cells from the bone marrow, alter their genomes, and then replace them in the patient. So the problem with sickle cell anemia, the gene that produces beta globin, a component of hemoglobin, has mutations that cause the protein to clump, and what that results in are these deformed shaped red blood cells. So the solution, the beta globin rescue. Scientists engineer a virus to deliver a functional copy of the gene that produces beta globin, and the gene has been modified to prevent sickling. So here's our engineered virus. It has the functional gene for beta globin. Put it into the patient's bone marrow. So the cells, the marrow that makes red blood cells now has the right gene and can make the proper blood cells. And I'm sorry you can't really see this. Even if I zoom in, you're not going to be able to see. So hopefully you can see it on your own paper or device, whatever it is that you're using. And now let's look at HIV. HIV stands for Human Immunodeficiency Virus. HIV. And this is a virus that's going to infect the helper T cells. And this is believed to have crossed from primates to humans in Africa way back in the 1920s. There are several strains of this that exist. And as they attack the helper T cells, this deletes acquired immunity. So a couple people have acquired immunity from HIV through a bone marrow transplant because the bone marrow donor had a mutation that prevented HIV from being able to infect their or bind to their helper T cells. So that's pretty interesting. If we could find people with this mutation and copy their genes somehow and use it for treatment or something and I don't even know if they're currently doing that, but that would be really cool. So the result is a decline in white blood cell population that leads to AIDS, and that stands for acquired immunodeficiency syndrome. And if you look over here, that's kind of the last category of HIV. So let's analyze this chart here. On the y-axis, we have T lymphocytes in the blood. So basically how many white blood cells you have. And then on the x-axis, we have years since infection. And, this first line is what's representing the white blood cells. And the second one is what's showing the amount of viruses. So you'll see there's the acute phase, the chronic phase, and then full-blown AIDS. So in the acute phase, there's a rapid progression of the virus. So upon initial infection, the HIV, the amount of virus in the blood goes way up super fast, but then you'll see almost immediately we've got a really quick drop. So initially your immune system is doing what it's supposed to do because you still have a high amount of those white blood cells and it's kind of fighting off that infection. So your body is producing enough T cells to keep up with the virus. in the chronic phase, the section right here, 3 to 7 years after infection, what you'll see here is you'll get symptoms starting. So during this time, the acute phase at the start, you're asymptomatic. That means without symptoms. So you could have HIV for up to 3 years and not even know it and be spreading it to your sexual partners and have no idea, which is what is scary about it. But some of the symptoms are diarrhea, shingles, fever, fatigue, cough, and I'm sure there are many more. And what you'll notice here is that the white blood cell count is starting to go down as the HIV virus amount in the blood goes up. So you're starting to lose your acquired immunity here. In this last section with AIDS here, the T cell count decreases drastically. You can see it go way down almost to nothing. And the virus continues to increase in the blood. So this makes the person susceptible to opportunistic infections like pneumonia or tuberculosis. So people don't die of AIDS. They die of something that their body, some other sickness that their body couldn't fight off because they have AIDS, if that makes sense. So if you're going to test yourself for HIV during this acute phase, like you're just getting a test for it, it's not going to show up until 25 days after infection. And if you are familiar with Freddie Mercury, he's one of the singers in Queen, I believe, the band Queen. He died from bronchial pneumonia as a result of AIDS because he didn't have the immune system or the white blood cells to fight. Just something that a person without AIDS would have probably been able to fight off and be fine from. And I believe there's nothing about COVID-19 on the exam, but we're just going to cover a couple of things about it real quick. So COVID-19 begins with an upper respiratory infection, and your upper respiratory system includes your nasal passages, your pharynx, your larynx, and your trachea. So basically your nose, your throat, your mouth, and the tube that leads to your lungs. And so what happens here is the COVID virus invades these cells. It takes them hostage and replicates. So most people's immune system can contain the virus within the upper respiratory system. Using the T cells and antibodies. But if it can't, it can progress into a lower respiratory infection, and that's where we start to see a lot of problems. So it targets your goblet cells, which, remember, those are the cells that produce mucus. So it's going to cause more mucus to be produced. And inflammation of your bronchial trees, the branches that go off into your lungs. Sometimes the body may kick in a severe immune response, which is going to cause more inflammation, and to the point where your alveoli, these little grape-like sacs in your lungs, can fill with fluid. This can lead to pneumonia. And this is typically when they put them on a ventilator. And the purpose of the ventilator is just to help the lungs expand and breathe while they can heal, while your body recovers. And then hopefully, if you recover, they can take you off the ventilator. So you can read through all the little details of this, but with severe COVID, this is what a healthy alveoli looks like. They're fully opened, they're clear, they can easily exchange oxygen and CO2. But when it becomes moderate, you'll see they start to fill with fluid and they're not quite as open, and so you're not going to be able to exchange gas as efficiently. And then with severe COVID, these are completely filled with fluid. You can see they're starting to collapse because it starts to affect the surfactant that helps keep these open. And gas exchange is greatly hindered here. So that's it for infectious diseases. And again, this is the last packet in this unit, or in this module. So we do have a test coming up. I will get a study guide review up as soon as I can, hopefully within the next couple of days. I'm recording this on Friday. So watch for that and then be ready to take the exam starting November 15th. Bye, guys.